Spotlight, lectures and performances on and around Albany State University.
Hello.
Hey, good afternoon. You didn't get lunch yet, right? So that's well, I'm very glad to introduce Dr. Raju. I call him Dr. Raju. His name is Thaugaraju, who is the assistant professor at the Georgia Health Science University. And I asked him to send his resume. He sent me a resume like this. It's 11 pages. So I'm going to read all of this. So I can spend about 20 minutes, then he can talk about 30 minutes, right? So he's actually served as my research mentor, and he published more than 50 published papers. And he's also served as lots of research reviewer at the NIH, NSF, like that. So he's very active in research. So this is my third year start our collaboration, and he's served as my research mentor also. So I'm very glad, and we talk about more our research to continue and writing proposals and writing papers, all kind of things. So I'm very happy to see him here. Then I like.
Thank you very much, and thank you very much for the invitation. And this is my second visit. And last year also I came with my chairman, Dr. Ganapati. And so today, I am going to talk about one of the transporter or the tumor suppressors. Many genes, it is downregulated or activated in the several cancer. But I am interested in one of the gene. So in my talk, maybe I will go in the two part. The first part, what is the molecular mechanism of the gene is silenced, and what is the importance of that gene in the mammary gland and the development. So here I will give you some brief overview what is SLC5A8. The SLC5A8 stands for solute transporter. Since the transporter means it transport something from the outside to inside the cells. So this is SLC stands for solute transporter family 5. There are 43 families and more than 500 transporter has been discovered. And this is the family 5, and the subtype is A8. It is also called as sodium monocarboxylate transporter 1 or SMCT1. It mainly transports a small chain fatty acid or SFA like acetate, propionate, and butyrate, and lactate, pyruvate, and nicotinate from the outside the cells to the inside. And it is also transport iodide, especially in the thyroid gland. And it is located in the chromosome 12, Q23.1. And it is a 68 kilodalton protein. It is highly expressed in the thyroid gland, and then it is in the kidney and the colon, and then mammary gland, brain, liver, and the prostate. And the SLC5A8 is expressed in this. There are two sides in the membrane. If we take the membrane, there is one in the basolateral and one is the apical. The basolateral side, it is towards the bloodstream, and the apical in the opposite side. So it is this particular protein is expressed specifically in the apical side of the thyroid gland or mammary gland ductal epithelium. And this is the first transporter, since several genes are inactivated or activated in the several tumor, but the transporter is not discovered as a tumor suppressor or oncogene, but this is the first transporter has been discovered as a tumor suppressor in several cancer. So the main function of tumor suppression, it is involved in the inhibition of histone deacetylase. Since there are if we take the DNA in the strand, there are two mechanisms. One is the histone acetylase. The histone acetylation is involved in the gene transcription. The histone deacetylation is involved in the suppression. So in most of the breast cancer, what of the tumor suppressor is inactivated by the HDAC, since it is unwind, to wind the DNA, so the DNA transcription cannot accessible for the transcriptional activity. So the gene, what it does, it transports some specific substrate into the cells, and thereby it inhibit the HDAC activity, thereby the DNA get wind, so the transcription machinery can take the tumor suppressor, and thereby it involved in the regulation of the tumorigenesis. So as I mentioned before, that gene has been discovered first, that tumor suppressor in the colon, but then it is discovered it is also inactivated in the thyroid gland and the kidney, stomach, brain, and we discovered it is inactivated in the breast cancer and the pancreas, prostate, leukemia, head and neck. This is the first tumor suppressor have been ever reported. This more than 10 different types of cancer. Whatever the tumor they checked, most of them it is down-regulated. So nobody knows why the transporter is specifically down-regulated by the tumor cells. So we are interested to find out what is the mechanism, why the transporter is down-regulated by the tumor cells. So this is just schematic representation, but it's everybody knows if you take a high fiber diet, it reduces the colon cancer incidence, but it is the mechanism is not discovered before. So we believe that when the SLC5A8, the transporter, might be involved in the tumor suppressor associated with the high fiber diet. If you take the dietary fiber, it converts into the small charge chain fatty acids like butyrate by the bacterial fermentation. So if you take the dietary fiber and it is fermented by the bacteria, and then it will release the bacteria and the butyrate as a byproduct, it is located in the outside the cells. So if in the SMCT1 or SLC5A8 express in the apical side of the membrane, it taken up this charge chain fatty acid, get into the cells, but it does the two ways. In the normal cells, the butyrate involved in the differentiation, it gives the energy and also involved in the differentiation, and then it is released from the blood side, the basolateral side, and involved in the immune cell regulation. This is in the normal cell scenario, but in the cancer cells, what it does, once if the SMCT1 or SLC5 get into the cells, it inhibits the HDACs, and thereby it kills the tumor cells specifically. It does not kill the normal cells. So the gene was first discovered in the thyroid gland. You can see the immunohistochemistry analysis. The gene or the transporter is expressed in the apical side of the thyroid gland, or this is the first report they showed that it is expressed in the mammalian cells. And then in 2003, a paper came out in PNAS. They were the one first discovered it act as a tumor suppressor. What they have done is they have taken several normal tissue, colon tissue, and the tumor tissue, and they found that in most of the normal tissue it expresses the transporter, but all the cancer cells it is inactivated. The same thing, they have taken several cancer cell line also on the colon cancer cells, and that the colon cancer cells also inactivated the function, and then they found that the transporter is inactivated by the DNA methylation. Since the DNA methylation is inactivated the several tumor suppressor. So to reactivate the gene, what they have done is they have taken the colon cancer cells and treated with the DNA methylase inhibitor like 5-azacitidine, and then they checked the expression of the gene, and once if the treat the cells with the 5-azacitidine, it is reactivated. So they found the mechanism. The transporter is inactivated by the DNA methylation. If you inhibit the DNA methylation, the gene getting reversed or reactivated in the colon cancer. These are all just the methylation and methylation PCR. So after that, so then they discovered that the SLC5A8 is also down-regulated in the colon cancer, and yeah, this is just overview. What is the function of the butyrate? Whatever the transporter is taken up, the butyrate from the outside to inside. The butyrate is already known. Butyrate, it is suppresses the metastasis and activates the p53, and it inhibits the anavikus by activation of the another gene CACAM, and also it is activated the death receptor 5 that is specifically in the tumor cells express the DR5. So in the butyrate activate the receptor by DR5, and thereby it induces trial signaling, and also activate the FAS mediated cytotoxicity, and these are all involved in the inflammation and oxidative stress. So they first discovered in the colon cancer it is inactivated, then they found that it is inactivated in the gastric cancer. I'm not going through all the detail how it is inactivated, since I will focus only in the breast cancer, and here just I am giving the overview how in what are the cancer it is inactivated. Here they took the thyroid cancer, and this is the wild type, and this is the normal thyroid gland, and this is the tumor. So the tumor cells it is inactivated, the expression, and also here they have taken the gliomas, the brain tumor cells, and here is the normal express the transporter in several of them. The tumor cells, the tumor tissue is inactivated, the transporter. And again the prostate cancer also it is inactivated, and the pancreatic cancer, head and neck, and acute myeloid. So these are, it is known that this transporter is inactivated in so many different kind of human tumor. So I have been working in the breast cancer, so I was interested whether what would happen in the breast cancer. So what I have done is I have taken the normal mammary tissue and also the mammary tumor, the same paired sample from normal and the tumor of 5 pairs, and we found that in all of them the tumor tissue it is almost completely 100% is inactivated, the tumor and the transporter. Then we have taken the normal mammary epithelial cells and the cancer cells, and all normal cells express the transporter, and the cancer cells it is completely inactivated. And this is the tissue array, the RNA array. It has several normal tissue at a different normal tissue, and the tumor at a different, like adenocarcinoma, tuckle carcinoma, and different kind of mammary cancer or breast cancer, and it is also inactivated when compared with the normal. All tumor tissue is inactivated. This is the quantification. You can see the normal express very high expression, but in the tumor tissue it is almostly gone. And this is the protein immunohistochemistry. You can see in the normal cells it is expressed, but in the tumor cells almost completely inactivated the protein. And this is the function of the transporter. This is measured by the nicotinate uptake. The all normal cells it show around 50% of the transporter, but it is almost inactivated in the tumor tissue. And then again what we have done is we have taken the normal mammary epithelial cells and the breast cancer cells and treated with 5-azacitidine. Since we inhibit the gene, it is already known that is inactivated by the DNA methylation. We treated with the DNA methylase inhibitor and checked whether it is reactivated. But in the normal cells, it is already expressed. Even if you treat with the azacitidine, it doesn't affect. But if you take the cancer cells, it does not express the SLC5A8. But if you treat with the azacitidine, it reactivated. Since again here the gene is inactivated by the DNA methylation. But if we inhibit the DNA methylation, it is reactivated. And then what we have done is we have taken the cancer cells. Since in the cancer cells it is specifically inactivate the transporter, what would happen if we reactivate or overexpress the gene? So we have taken one of the breast cancer cells and re-expressed the transporter. And then we treated the cells with one of the substrate, the pyruvate. Since the colon has a lot of butyrate, since they are taking the high fiber diet and the bacterial fermentation, it release the butyrate as a byproduct. But the mammary gland is not butyrate. The another substrate is a pyruvate. The pyruvate is ubiquitously expressed in the bloodstream. So that's why we use the pyruvate and treated the cells in the present absence of the transporter and treated with in the present absence of pyruvate, and checked the cell death using the FACS analysis and show that if you put the pyruvate in the presence of SLC5A8, this cell death occurs. This is cell cycle, and this is a specific annexon labeling. It is specifically distinguished between the necrosis and the apoptosis. This whatever the cell death occurred by the butyrate, it is due to the apoptosis. And these are all the marker for the apoptosis, the PARP cleavage. And so we looked for the mechanism how it is involved in the apoptosis, and checked several genes by real time in the RT-PCR, and this is the protein. And it showed that the gene is activated through the trial signal. There are three death receptors: the FAS, TNF, or the trial. Those are the three death receptors involved in the apoptotic signaling, but the pyruvate specifically go through the trial signaling by the activation of trial and the death receptor. And here again we it is already known that the function of the transporter is inhibit the HDAC. What we have done is we have taken the breast cancer cells and express the transporter, and incubate with the different concentration of the substrate of the SLC5A8, butyrate, pyruvate, propionate, and acetate. It shows that the butyrate has a high effect. It inhibits the HDAC activity. Then the pyruvate and the propionate, the acetate and the lactate does not inhibit the HDAC activity. So it may be, and the inhibition of HDAC activation is also measured in the acetylation of the histone. You can see in the presence of pyruvate, it reactivated or induced the acetylation, and the butyrate propionate also it does, but the lactate or acetate does not induce the acetylation. And we did the colony assay in the absence of SLC5A8 and also the different substrate. Once if you put the vector control, all substrate doesn't have any effect, but if you put the transporter, the pyruvate, butyrate, and the propionate inhibit the cell the colony formation assay, it shows that this substrate in the presence of SLC5A8 induced cell death. So this is again, and since the pyruvate kill the cells, since there are 5 isoforms the lactate dehydrogenase in the human, the LDH 1, 2, 3, 4, and 5. The LDH 1, what it does, it convert the lactate into pyruvate. The LDH5 converts the pyruvate into the lactate. So we checked in the normal and the cancer cells with the LDH1 and the LDH5. You can see in the LDH1 is specifically expressed in the normal cells, but in the tumor cells it is completely inactivated. So the same thing, the opposite, the LDH5 is expressed only in the cancer cells, the normal cell doesn't happen. So which means the cancer cells need more lactate to, since the lactate has the acidic function. So once the tumor cells convert the pyruvate into the lactate, the tumor cells can get more lactate, and that lactate can be used to proliferate the adjacent tissue. For if one cell is tumorigenic, then how it invade to the other cells, how to kill the other normal cells, they use the lactate to acidify the cells. So if the another cells get more acidification, the cells automatically will die. So the tumor cells can evade, and thereby it is go for the one mammary gland to another mammary gland, or the distant metastasis and all. So it is well studied in the role of LDH5, the LDH1, the LDH5 is the oncogene and the LDH1 is the tumor suppressor. And also we have checked the HDAC activity in the normal cells and the cancer cells. You can see normal cells very low, and the cancer cells express very high HDAC activity. The same thing, the acetylation, where normal cells has more acetylation, but the cancer cells very low. So this is just a schematic representation how the normal cells and the tumor cells use the transporter. So if you take the glucose get into the cells by the glucose transporter. The glucose is converted into the pyruvate, but in the normal cells the LDH5 is low expressed. So it is a very low amount of pyruvate it converted to the lactate, but the LDH1 is highly expressed. So it is other way, most of the lactate is converted to the pyruvate. That pyruvate get into the mitochondria, involved in the metabolic regulation and also inhibit the HDAC activity. Here the SLC5A8 also transports the lactate, so more lactate it is converted to the pyruvate and involved in the energy metabolism. But in the cancer cells, you can see the transported, the same thing, glucose is taken up and get into the cells, and the glucose is converted into the pyruvate, but the LDH1 is LDH 5 it is highly expressed, but LDH1 is low expression. So most of the pyruvate is converted into the lactate, but whatever the accumulated lactate, it is taken up by the another transporter MCT1 to the outside the cells, thereby it is kill the nearby normal cells. Yeah.
This mechanism does not contradict. We know the cancer cells, they grow very fast and they need energy, and this is one of the pathways for the production of energy. How they convert part of this pyruvate into lactate was not going to reduce the energy source?
Yeah, that is the energy source. It is since if we go for this way, I think the cells can get around 30 to ATPs. So if we go to this way, it will get 2 molecule of the ATP. So that's what in most of the cancer cell, it has the more glycolytic signaling pathway. More and more glucose get into the cells and more pyruvate to produce to compensate from 1 molecule of 32 molecule of ATP, and here the cells will get only 2. So 16 times they need more ATP. That's what they increase the more proliferation. That's when the cancer cells grow much faster to take the energy. And if you put the transporter again, so the same mechanism occur. The glucose is converted to the pyruvate and the lactate, but if you put the transporter, it increases the LDH1 and the conversion of pyruvate to lactate is much decreased, and also it is inhibit the HDAC activity and induced apoptosis. That is just schematic representation of the function of the transporter. So then we wanted to know what is the mechanism of the transporter is inactivated in the breast cancer. So what we have done is we have taken the normal mammary epithelial cells and put the several oncogenes which involved in the cellular transformation like H-Ras, c-Myc, constitutively active STAT3 and BCL2 and ETF1, and then checked the SLC5A8 expression. If you see, you can see the constitutively active H-Ras, it inhibit the expression of the SLC5A8, but the c-Myc also does, but not when compared to the H-Ras, and the c-Myc has some lower significant effect, but the constitutively STAT3 or BCL2 or ETF1 does not affect the expression of the SLC5A8. That is the RT-PCR, and this is the real-time PCR, shows that the H-Ras inhibit much efficiently the expression of SLC5A8, but when compared to the other oncogene, and this is the protein, and we have also measured the methylation specific PCR. This shows that maybe the H-Ras, constitutively active H-Ras might involve in the inhibition of the transporter. And the H-Ras, we also wanted to know whether the DNA methylation involved in the inhibition of the transporter, whether we wanted to know any of those, the oncogene does affect the DNA methyl transfer. There are 3 DNA methyl transferases, DNMT1, 3A and 3B. So we checked the expression of DNMT1, 3A and 3B, and in the absence of oncogenes, we can see in the H-Ras specifically activate the DNMT1 and the TNMT3B in some extent, but the c-Myc also it is activate only the TNMT3B but not the TNMT1. Again, this shows that the H-Ras activate the TNMT1, and the TNMT1 maybe it binds into the SLC5A8 promoter, and thereby it inhibit the function of the SLC5A8. That's what we, that is the RT-PCR, and this is the real-time PCR. And then what we have done is we have cloned the TNMT1 promoter, whether the H-Ras might activate the DNMT1, that is the RNA level. We wanted to know what would happen at the promoter level. What we have done is we have fused the DNMT1 promoter with the GFP construct. If any gene would activate the DNMT1, you can get the green fluorescent. If gene doesn't activate the promoter, you may not get any green fluorescence. So this is the vector, and if you put the oncogenic H-Ras, you can see more green fluorescence, which means it activates the DNA function. But the c-Myc also it does little, but the STAT3 also not like H-Ras, it has some effect, but none of the other oncogene involved in the activation of the fluorescence. And this again is the activity measurement fused with the luciferase. You can see H-Ras has dramatic effect activation of the luciferase when compared with the other oncogene. And then we wanted to know whether the DNMT H-Ras DNMT1 form a complex, and thereby it inactivated the SLC5A8. And we did the chip assay. What the chip assay we have done is we have transfected all the oncogene and immunoprecipitated the DNMT1 antibody, and specifically did the PCR for the SLC5A8 promoter. You can see only the H-Ras form a complex with the DNMT1, and somehow the c-Myc also form a complex in the two sides of the SLC5A8 promoter. Again, it shows that the H-Ras DNMT1 form a complex, it inhibits the SLC5A8 function. So that is the transient expression. So we wanted to know what would happen if we express stable the H-Ras or oncogenic H-Ras in the cells. So we have again taken the normal mammary epithelial cells, and we developed the overexpressed oncogenic H-Ras, and this is the transformed cells, and we injected into the mice, and it formed a tumor. And that is the primary tumor. We isolated the cells from the tumor, and we left it for a longer time, waited for maybe 4 months, and it formed a lung matter. Whatever, if we injected the mammary fat pad, it formed a mammary tumor, and then it is metastasized to the lung. So we also isolated the cells from the lung. So here from the normal, and this is transformed. This is the primary tumor, and this is the lung metastasis. So we collected the series of this transformation from normal to metastasis, and checked. This is the H-Ras activation from the western blot. You can see the activation of the H-Ras, and then we checked the promoter. You can see at the mRNA and the protein. Once the cells get transformed itself, the transporter is inactivated. But in the primary and the metastasis is completely gone, both mRNA and the protein level. And this is again the real-time PCR, and this is at the function, and this is the methylation. Once if you put the H-Ras, it induces the methylation. Then we have done using all 4 cell line with the microarray, and it shows that the SLC5A8 is highly downregulated gene. It is if you put the H-Ras alone, it is around 80-fold. It is downregulated in the normal cells. That's what it showed. It is completely gone. And this is again the how the H-Ras inactivate the function. It is activation of the DNMT, whatever we have seen before. And this is again versus the normal versus the primary tumor, and here also the SLC5A8 is the highly downregulated. When compared to the normal versus metastasis, again it is around 70 to 80-fold. It is inactivated the function. So that is in the cell type. So what would happen if you take the mammary tumor itself? So what we have done is we have developed the spontaneous mammary tumor mice using the MMTV-HRas. And this is the wild type, doesn't have normal mammary gland, and this is just expression of the transgene alone, the MMTV-HRas. And if you leave it for 1 year, it form a spontaneous tumor. So we collected the tumor, and again checked the HRas expression. This is showed the transgene, and the tumor it expressed the HRas. And at the same time we collected 3 different mice, normal, just the transgene alone, and the tumor. You can see when compared to the wild type, the transgene itself it is reduced significant of SLC5A8, and the tumor it is almostly downregulated in all 3 tumor, the mRNA and the protein, and also it is a methylation. Then what we have done is we have what would happen in the stem cell level between the normal, the transgene, and the tumor. So we isolate the stem cells between the normal tissue. So this is the stem cell. This is the CD49F, and the CD24 is the marker for the mammary tumors, mammary stem cell. So this is you can see the CD49F high and the CD24 low. This is the stem cell, and this is the progenitor cells. And the normal cells is around 9%, but if the transgene, it is increased to 19%, but in the tumor it is close to 35%. But you can see the normal mammary gland, you can 3 different population. This is fibroblast, progenitor, and stem cell. But once that transgene occurs, most of the normal cells are the progenitors. It is converted into the stem cells, but in the tumor, it is almost all of the progenitor, and you can see only one population. It is missing the progenitor and the fibroblast. All of them is converted into the cancer-inducing or cancer-initiating stem cell or the cancer stem cells. And this again, we did the mammosphere using the wild type and the transgene. Under the tumor, the mammosphere formation is also increased depends on the transgene and the tumor, and that this is the just a quantification. And this is again, we checked the since the transporter is inactivated in the tumor, we wanted to know what would happen that TNMT1 expression between the normal transgene and the tumor. You can see in the normal very low expression, whatever the red, it is the TNMT1, and this is the marker for the stem cells CD49F. And if you colocalize, and this is the DAPI, since the DNMT1 is the nuclear protein, and you can see in the transgene, whatever the purple or the color, it is the colocalized is the DNMT1, and the DAPI, it is much low, where in the normal it is almost undetectable, but in the transgene it is increased little. And in the tumor, you can see several, the TNMT1 expression or the localization is much higher. And also we checked the DNMT activity increased from normal to transgene under the tumor. Again, it shows that the HRAS, not only in the cell type, in the in vitro, in the in vivo also, in the spontaneous mammary tumor, the HRAS, if you put the HRAS, it activates the DNMT1 and induces the stem cell, and thereby, or it inactivates the SLC5A8, thereby the cancer initiating stem cell number is increased. And then what we have done is, so it is in the in vitro, the in vivo itself, it is the MMTV-HRas inactivate the SLC5A8. So what we have done is we have generated the knockout mice of the transporter, and then we crossed the knockout mice with the MMTV in the spontaneous mammary tumor, and we checked the tumor incidence between wild type and the loss of one allele, the heterozygous, and the homozygous. And here you can see the tumor incidence, the spontaneous tumor, around 60 out of 36 mice, 23 form in the wild type, around 64%. But the deletion of one allele in itself, it is increased around 15% more, 75. But if you knock out both the allele, it is around 90% of the mice develop the mammary tumor. And also in the tumor formation time, it is around 210 days in the wild type, but the knockout, no, the 289 the knockout, the wild type, the knockout is around 216 days. And also we counted number of tumor when compared with the wild type and the knockout. In the wild type, number of tumors, one is higher in the wild type, but in the knockout is low. But you can see the more than 3 tumor, it is in the knockout mice when compared to the wild type. So which means the tumor incidence is increased on the early onset of the tumor, and the number of tumor also it is increased in the knockout mice. And also the tumor size is dramatically increased when compared to the wild type and the knockout. And this is just the K67 to check the cell proliferation. This is the SLC5 wild type and the knockout. You can see more cellular proliferation in the knockout. And also we checked now how many mice it is went for the lung metastasis, but in the wild type, only 17% of the mice went for the lung metastasis. But if you knock out the gene, around 42% of the mice went for the lung metastasis. And this is again the lung metastasis section. You can see more proliferation in the lung metastasis in the knockout mice. And also checked the survival time. The normal mice survive around 450 days on average, but in the knockout mice only 309 days on average survival. And also we collected the tumor between the wild type and the knockout mice and screened what are the genes it is affected, and we found several genes, but this is the mostly upregulated genes, the FGF, the growth factor. Most of them it is involved in the tumor genesis. The ANGPTL4 is specifically involved in the lung metastasis. Those genes, it is highly induced in the normal SLC5A8 knockout mammary tumor when compared to the wild type. So that is, if you knock out the gene, it is increase the tumor incidence, early onset of the mammary tumor, and also the increase the lung metastasis. So what we wanted to know, what would happen if we re-express the same gene in the mouse? So we made the mammary gland specific transgene. So we put the SLC5A8 in the mammary gland specific using the MMTV promoter, and this is the just a schematic diagram how to make the animal, and this is the genotyping. You can see that we made the mammary gland specific SLC5A8 knock-in mice, and this is to just check the expression between non-transgenic and this transgenic. The expression SLC5A8 is increased. Then we again crossed the transgenic mice with the MMTV-Neu mice. You can see if you constitutively express the incidence of the tumor is dramatically, almost 50% is reduced, and the number of days and the formation also when compared with the non-transgenic, the transgenic is delayed the mammary tumor formation, and the number of tumor is reduced, also the reduce the tumor size and the lung metastasis, and increases the survival time. So far what we have done is the gene is inactivated in the breast cancer by the DNA methylation. In the in vitro, in the cell culture system also, if you overexpress the gene, it is induces apoptosis, and also if you overexpress the oncogenic H-Ras, it is inactivated the gene by activating the DNA methylation, and if we knock out the gene, it is very aggressive to the tumor formation, induces lung metastasis. If we constitutively active the gene which we to protect the tumor genesis. So just this is the summary of what we have done so far. So then the second part, we wanted to know what is the specific function in the normal mice, what it doing in the normal, since that is so far it is necessary for the prevention of the tumor genesis. The next part we wanted to know what is the normal function of the transporter in the normal mammary epithelium. So this is just the schematic diagram how the mammary gland developed. So the mammary ducts are established in the fetus itself. Once the puberty, the mammary tuckle is established, during the pregnancy, the mammary epithelium is increased more than 300 times to secrete the milk. So once the lactation occurs and after the birth, the baby suck the milk. These are all the milk producing glands. So after some time, the gland has to go back to the pre-pregnant state. So several mechanisms is involved in this process, or here also from the mammary gland development. And since from here to there is a lot of cell proliferation, and from here to there it is a lot of apoptosis. The people are considering from here to there it is like a oncogenesis, the tumor genesis. From here to here this kind of a tumor suppression. So we looked in both ways, what is the role of that gene, where it is specifically expressed. So those are the genes which involved in the regulation of the mammary tumor, and also it is involved in the regulation of the involution. So what we have done is we have collected the mammary gland from the virgin and a different time of pregnancy and lactation and the involution, and the gene is specifically is activated in the early onset of the involution. So from that the phase which occur a lot of apoptosis, it is from here to here the gene is highly expressed, not from here to there. And that is the mRNA, and this is the real time, and you can see that gene is specifically involved in the apical membrane of the mammary epithelium. That is the SLC5A8, that is the basolateral marker, and the DAPI, and merged. You can see that is specifically expressed in the apical. And this is, again, we used some positive marker. They already known the STAT3, and also we don't use it as a positive marker. It's specifically involved in the involution process. So again, here we have taken that SLC5A8 wild type under the knockout. This is the wild type and the heterozygous and the knockout. And what we have seen here, we have collected the normal mammary gland from the wild type and the knockout. When compared with the wild type, you can see the normal more branches, the hyperplasia occur in the normal on the SLC5A8 knockout mammary gland. And what we have done is we have isolated the mammary, the epithelial cells from the wild type under the knockout, and just put it in the petri dish and stain with the GIMSA, which only the live cells stain the GIMSA. And it shows, and we can also quantify how many cells in the dye binds in the cells. When compared to the wild type, the knockout cells, the knockout mammary gland is more mammary epithelium, and you can see both the GIMSA and the activity. And also we have isolated the mammary stem cells from the wild type and the knockout. This is the fibroblast, progenitor, and the stem cells. Here you can see in the normal cells around 1% of stem cells, but in the knockout mice, in the stem cell, it is highly increased, around 4-fold. It is increased stem cells. And this is just we developed the mammosphere to check whether it is proliferate or differentiate the mammary stem cells, that is the wild type and the knockout, and stain with one of the dye. It is the dye which binds to the membrane, so that once the cell divide, that the dye is passed on to the daughter cells, which means if the cells grow much faster or divide faster, the dye will get diluted, and after sometimes it disappear. If the cells divide very slowly, the cells retain in a longer time. So that's what it happened in the normal cells. It is divide very slowly, so you can see the intensity of the dye. You can still after 3-4 passage you can see the dye, but in the knockout cells, because it proliferates much faster, so the dye is getting diluted from the passage from the mother to the daughter cells. That's what which means increase in the proliferation with the dilute the dye. Again, it showed that the hyperplasia occur in the gland. And then we checked for the morphology between the wild type and the knockout. Just this is the at the wild type and the knockout. We checked the expression of the SLC5A8 at a different time. And here you can see the morphology. So if you take the cells at involution day in the wild type, you can see a lot of the brown dye, which means the live cells, more epithelial cells. Whatever that the white cells, maybe most of them are fibroblast. Here you can see on the day 2, most of the milk lab or milk particle, but at day 3 and all, the milk is ready, since there is no suckling. So how we did the involution after delivery, we left the mice for the pups for 10 days and then remove the pups. So once if you remove the pups, there is no suckling, so the milk get accumulated. So because of the pressure, the accumulated milk induces the lot of epithelial apoptosis, the cells will die. So that's what we induces involution, and collected the cells at a different time point, and you can see at the involution day 4, it is almost the involution process is completely finished or completed in the wild type, but in the knockout cells you can see more the brown cells, which means more live cells. It is intact mammary gland. It is still doesn't go for the involution. It is getting delayed. But after 8 days, you can see it is regressed. But maybe there's some other compensatory mechanism. But at 4th day, you can see a definitive involution delay, which means that apoptotic signaling is much reduced in the knockout mice. And these are all just the marker for the involution. It is already known STAT3 phosphorylation in the wild type and the knockout, and we checked some of the specific genes which involved in the involution. When compared with the wild type, the knockout, the expression is much reduced, which these are all involved in the apoptotic signaling. And here also to check the apoptosis, we check the staining, and whatever the brown staining, the apoptotic cells in the day 1 there is not much apoptosis. On day 2 you can see wild type started apoptosis but not in the knockout mice. On day 3 you can see more brown staining the wild type but not in very low in the knockout mice. But at day 4 it is the process is almost completed. There is no cells has to go for the apoptosis in the wild type, but still the knockout the apoptosis started. And this is again to show the HDAC activity, because the gene is involved in the HDAC activity inhibition. We checked the HDAC activity, and there are so many HDAC, there are 18 members that have been identified, but we checked the HDAC 1 and 3 expression, which is specifically involved in the butyrate dependent apoptosis. When compared to the wild type has low when compared to the knockout in the wild type has relatively low HDAC expression in all time point, and this is the HDAC 1, HDAC 3, and the activity also it is increased, and this is again the acetylation. And also what we have done is we have taken the normal mammary epithelial cells and express the SLC5A8. Since these are all the intact mice, what would happen the normal mammary epithelial cells, and since if you take out the mice after 10 days, the milk get accumulated, but nobody knows how the accumulated milk induces the epithelial apoptosis. So there are two mechanisms. Since the milk accumulation will induces the mechanical stretching, so the lot of pressure, so that pressure may induces the apoptosis. The other thing, whether something in the milk can induce apoptosis. That's what we have done is we have taken the normal mammary epithelial cells and set up the mechanical stretching, which is same kind of pressure when the milk accumulates in the mammary gland. And then we have treated the butyrate. Since the breast milk contains appreciable amount of butyrate, when in the next colon the breast milk contains the butyrate. So that's what we have taken the normal mammary epithelial cells, did the stretching, and treat with and without the butyrate. If you do the stretching alone, you can see that the apoptosis is not induce any apoptosis. But if you put the stretching under the butyrate, the apoptosis is dramatically induced in the wild type mice. But if there is no SLC5A8, it cannot transport the butyrate. So it even though if induces the stretching, it cannot induce apoptosis. What would happen if you remove the pups? It accumulates milk, induces pressure, and that pressure itself it is activate the SLC5A8 expression, and that SLC5A8 induces more and more butyrate from the milk, and thereby involved in the apoptosis signaling. And this is so for the intact mice, we do not know whether SLC5A8 necessary for the induction of the apoptosis in the absence of butyrate. What we have done is we have taken the wild type and the knockout mice and implant the butyrate. We have a tablet for 90-day release. Every day they release some 100 micromole of the butyrate for 90 days. And then we set up the breeding and collected the mammary gland after 10 days weaning, and collected the mammary gland at day 1, day 2, day 3, and day 4 of involution. You can see when compared with the wild type mice with this is the wild type mice, no butyrate, and this is the wild type mice with the butyrate, and this is the SLC5A8 knockout mice without butyrate and with the butyrate. And when the day 2 itself, it is when compared with the knockout mice, the wild type mice go for more apoptosis. But if you implant the butyrate, you can see the more and more the brown cells it is getting reduced, which means more cell death, and that epithelial cells is replaced by the fat cells. And you can see the clear difference on day 4. And day 4 still when compared to the day 2 in the wild type, it is go for apoptosis, but if you implant the butyrate, more apoptosis goes on. So this shows that the SLC5A8 is necessary to transport the butyrate present in the milk, and that butyrate is specifically involved in the apoptosis. These are all just to check the apoptosis and the HDAC activity. Yeah, again here we crossed the SLC5A8 knockout with another spontaneous mammary tumor. We developed MMTV-Neu, and we crossed again here checked the tumor incidence between the wild type and the knockout. Again around 50% mice go for induced mammary tumor, but here you can around 80% induce apoptosis. The same thing, early onset of the mammary tumor. Here you can see the lung metastasis when compared to the wild type. The knockout mice has more lung nodules, and also the percent here is 60% go for lung metastasis, but here it is around 85% go for the lung metastasis, and also the tumor volume, it is when compared to the wild type, the knockout, the tumor size is bigger and reduce the longevity or the survival time. And these are all just to summarize how what is the importance of the gene. So during the involution, the milk getting accumulated, that milk contains the butyrate. So that butyrate get into the cells by the SLC5A8. Once the butyrate get into the cells, inhibit the HDAC and induces the trial-dependent apoptosis, and thereby it reduce the here also here it is what does it mean? It is induces or the involution process. So that the involution process, the mammary gland remodeling is necessary for the prevention of the breast cancer. What we believe that since it is already known that the number of increasing the pregnancy reduce the number of the breast cancer in the human, but nobody knows what is the relation between the increasing or the number of delivery or increasing number of pregnancy reduce the reduction in the pregnancy. What we believe that, so every time getting the pregnancy, so the transporter is getting increased and it transport more and more butyrate and induces the differentiation, and also whatever the tumorigenic cells developed in the early stage of the tumorigenesis, that the butyrate might kill the tumor cells. So increasing the pregnancy every time, the same thing happen again and again. That is one of the mechanisms we believe that number of pregnancy related to reduced the incidence of pregnancy, but still we are validating using the right that is the next project we are doing, whether this gene is necessary or associated with the increased pregnancy and associated tumor suppression. And again the same, to summarize, the SLC5A8 expression is induced in early onset of mammary gland involution, and it is knockdown of the gene, it is involved in the hyperplasia, and it is delayed involution, and SLC5A8 involved in the HDAC inhibition, and the SLC5A8 inactivation predisposes the tumorigenesis. So most of the work is done by the Selva and the Sabarish, and most of the stem cells work done by my graduate student Rajneesh, and the Butyrate Prasad and Dr. Ganapati, our chairman. They helped me in all the aspect of the project. And this guy from Max Planck Research Institute from Germany, he developed the knockout mice, and these are the funding source. Thank you.